got to share my screen. Azeri, can you help me? How do I get this done? Yes, here. Yes.
Can you please, should I share screen your PPT or?
Yes, please. Do you have my PowerPoint?
It's not PowerPoint, it's PDF. Is it okay?
Let me see if I can.
Wait a minute.
Now?
Wait a minute.
Partager l'écran. Yes. Okay. Now. Yes.
Oh, yes.
I'm sorry about this, my dear students. Ah, Miss Dr. Afif is here. Hey, hey. Bonjour, Afif.
Bonjour, Zine. Oui, j'étais là depuis première. Hein. Depuis
Très
le début.
bien. Bonjour à tous les étudiants. Good morning to all students.
Très bien.
So we are going to deal today with the screening of recombinant bacterial colonies. Okay. Right. Can you see the screen now, all of you?
Yes.
Okay,
Yes.
thank you. So last time we focused on methods to clone prokaryotic genes and eukaryotic cDNAs, right? And so that they could be inserted into vectors so as to allow us to work with them. Work with them is to characterize them. And what I would like you to understand is the difference between a gene and a cDNA, right? You need in prokaryotes, in eukaryotes especially, right? This is very, very important, okay? So after having got the cDNA or the gene, inserted it into a vector, right? Today we are going to learn how to insert it into a vector, into a plasmid, then get it into a bacteria so that we can, don't worry for now, we will speak about all this, right? How to select on antibiotic and a special medium so that we only get the good potential candidates. And how do we recognize the real candidates that are of interest to us? So how do we get the positive clones we are looking for? So this is what we learned last time. We have a bacterium with a plasmid, and within this plasmid, we are going to insert a gene when it is a prokaryote, or a cDNA when it is a eukaryote, right, into this plasmid. So you will have cDNA banks or genomic banks. And all these will be put into bacteria, into something that we call bacterial transformation. We saw this last time, okay? But now the problem is, how do we know which ones are good? Because when you insert a gene sequence, right, Some of them are inserted. Some of them are not inserted. And some of them don't have any plasmids. Okay, so we need to have only this population, and within this population, only the ones that carry the gene that we are looking for. Okay, so to this end, we use two tools to screen among these three populations. We use the tool called selectable marker genes, and the second one is the reporter genes. Sorry, Azeri, do the students have the PowerPoint?
Not yet. We will share it after the lecture and
Okay, okay, good. But they have it on their screen.
yes.
Okay. Students, when you don't understand something, please interrupt me. Okay. So the challenge is to find the colonies that are carrying the sequences that we want to work on, that we want to target. And there are two most useful tools that you need to understand, first of all, and remember. The first ones are called reporter genes, right? And they are based on color differentiation. The reporter gene is a gene sequence, right? A DNA sequence that will code for a protein that will be responsible for a change of color. We will see this. And the second one is our selectable marker gene. Selectable marker genes offer a selectable advantage. These are, for example, antibiotic resistance genes. As there is somebody raised her hand, I think. I don't know who it was.
Apakah ada yang raise hand tadi?
Okay, right. So two things you need to remember: reporter genes and selectable marker genes. This will be used throughout the whole of your studies, right? And so you need to understand this, okay? Right. Voila. Okay, first thing to remember, we saw it last time, is that bacteria contain plasmids. So you need to learn this plasmid definition by heart and understand this. And there are two main features that will be relevant, that will be important to us today, is that these plasmids carry genes for resistance to an antibiotic. This is the first thing. And the second thing is that they have multiple cloning sites, that is, sequences recognized by restriction enzymes. Okay, I understand that you cannot see what I'm talking about, but it is important that I put this so that afterwards, when you go back to your lecture, you have the main essence of things that you need to remember. Okay, so reporter genes. Reporter genes are genes that will encode an enzyme that will result in a color change, right, in a substrate. So when you give this substrate with this extraordinary difficult name to remember, you don't need to remember this, but please just remember the X-gal, right? So this is the substrate, which is an analog to galactose, right? X-gal will be converted to lactose, sorry, will be converted to by beta-galactosidase to galactose and a product that has a very complex name that we call X. And this X is a blue precipitate. So therefore, the bacteria that have this, that are being cultivated with X-gal, right, will be converted, right, to X, and X will be blue. So therefore, the colonies will become blue. Okay? Right? So what you need to remember is that here you have a substrate called X-gal. It will be converted by beta-galactosidase. We saw this enzyme last time in the lactose operon. This enzyme will convert X-gal, will cleave to give you galactose plus X. Okay, so it will cut here. And the X will be blue. So therefore the colonies will become blue. So let us have a look on how it looks. So screening for recombinant colonies using reporter genes. We spoke about the lactose operon last time when we spoke about regulation of gene expression in prokaryotes. So you have the Z, the Y, and the A gene. The Z codes for beta-galactosidase, right? That's the enzyme. So what the researchers did, molecular biologists, they removed the 5' end of the Z gene, right? And they inserted it into a plasmid, okay? And this sequence is called the alpha peptide sequence. This means that this deleted 5' Z gene will encode for a peptide called alpha peptide. Okay? So, and on top of it, the researchers have inserted a multiple cloning site. So, multiple cloning site contains a stretch of restriction enzymes. We saw it last time. Restriction enzymes are enzymes that will cut specifically at DNA sequences, double-stranded DNA sequences. Okay? So, this is the basic structure. So you have, if this is the bacteria, right? The big blue rectangle. You have this engineered plasmid. And on top of it, you have the chromosomal DNA of the bacteria, but with a deleted LAC-Z gene. So you have here a short version of the LAC-Z and the portion that has been removed here has been put on the plasmid. Okay. So when these two, alpha is present and Z is present, and that there is no sequence in the multiple cloning site, this alpha peptide that will be translated will complement the 3' end of the Z gene, and the protein will be formed. Therefore, the galactose, the X-gal can be cleaved, and you will get a blue color in the colonies. This is a cartoon of a bacterial, of a petri dish with blue and white colonies. So the blue colonies, there is no sequence inserted within the multiple cloning site. In the white colonies, there is a sequence that has been introduced within the multiple cloning site, and this has caused a disruption of the alpha sequence, and because it is disrupted, it cannot complement the 3' end of the Z gene, and therefore there will be no active protein, and therefore you will not get any blue color. I sense that this is a little bit difficult. Yes or no? Can I have some feedback, please, from the audience? Hello, are you still there or are you all sleeping? I think they are dead. Everybody's here, but nobody's responding. So, Azeri, should I go through this again or not?
Yeah.
Yes.
Let me continue and then we'll see, okay?
Okay, you can continue. Yeah.
Right. So, first thing is report a gene. The second tool, important tool, is called selectable marker gene. A selectable marker gene will give an advantage to the cell, right? So for this, to this end, you need to remember the constituents of the plasmid. So in this plasmid, how do I get rid of this? So this is a typical plasmid. You have the origin of replication. Here you have AMP-R. This is a gene that will encode for resistance to ampicillin. Ampicillin is an antibiotic, okay? And here you have a multiple cloning site, this one, a set of restriction enzymes that has been inserted within the LAC-Z alpha peptide region in such a way that the alpha peptide open reading frame is not disturbed. Okay, so this is the normal setup of this engineered plasmid. You have the origin of replication, you have a gene coding for resistance to an antibiotic, a multiple cloning site inserted within the alpha peptide coding region. So, why do we need antibiotic resistance gene? Antibiotic resistance gene will code for a protein that will, so normally antibiotics, right, are lethal. They will kill cells. Okay, this is not concerning humans, but normally for bacteria, it is the case. If you don't have this antibiotic resistance gene, right, in bacteria, the bacteria will die. So when you do have the antibiotic resistance gene, the bacteria will survive because this will be coding for a protein that will stop ampicillin from being lethal. So if we insert something, a cDNA sequence within this gene, the gene will be disrupted, and therefore the antibiotic, therefore the bacteria will become sensitive. So this is a very, very important concept. We need to keep this because we are going to use it when we clone a foreign sequence within the multiple cloning site, right? So we saw this slide last time. So this is why I use the same slides. Okay. So once you have got your different sequences within plasmids, because you have introduced it, you have cloned it into your plasmid by using a ligase, you inserted it, and then you will use this to transform, to insert it into bacteria. So you will have here, you can see that you have a population of plasmids carrying different foreign sequences, okay, from the gene sequences you want to clone. And you have another population of plasmids that has not taken any gene sequence, foreign sequence. And so therefore, when you will insert this into bacteria, you will get different classes of populations. Bacteria carrying no plasmids, bacteria carrying plasmids with no inserts, bacteria carrying plasmids with some inserts, and bacteria carrying plasmids with the sequence we want. And this is the bacterial colonies that we want to select. And therefore, when we transform the bacteria, we're going to use the two properties I spoke about: selectable marker genes and reporter genes. So selectable marker genes, we're going to select the bacteria on ampicillin containing medium. Therefore, only the bacteria that will have the plasmid with antibiotic resistance, only these will grow, right? And then you will screen for blue or white colonies, and you will reject all the blue colonies, and you will keep only the white colonies. So this is the strategy that is used to select, to identify, to screen, so to eliminate all the clones we don't want. This is a selection procedure. We've got thousands of bacteria. Maybe we have only 100 recombinant colonies. And within the 100 colonies, we may have only 10 colonies that are carrying the genes we want. So this is a very, very difficult work that we have in front of us. Okay. So I gave this slide last week to you, but I'm giving it to you again so that you can see again that the question is about transferring a gene into a plasmid and using this recombinant plasmid to get into a bacteria. And therefore, once you would have screened your recombinant bacteria on selective medium and on X-gal containing medium, you will hopefully eliminate 90%, and you can only work with 10% remaining ones. Okay, so this is about chromosome library. It is always the same thing. You integrate bits. Oh mon Dieu, mon Dieu, mon Dieu, mon Dieu. Qu'est-ce que je fais? Voilà. So you integrate bits of chromosomes randomly into a population of plasmids, and therefore you will get a population of recombinant bacteria. And you have to have tools to identify the potential ones. And the two tools I'm talking about to you today is 1, selectable marker genes, and 2, reporter gene. So this is with eukaryotic. We start from the messenger RNA. It's always the same thing, right? Except that you need to be aware that the method is not the same whether you're working on prokaryotes or eukaryotes. Okay, so what do you get in the end? You get recombinant. This is a very nice experience that has worked very well because there are no blue colonies, there are only white colonies. This means that all these colonies have integrated a foreign sequence. Why are they white? And they grow on ampicillin. So if they grow on antibiotics, and if they are white, this means that the sequence that has been inserted has disrupted the alpha peptide sequence, and therefore there is no complementation with the remaining ligase chromosomal DNA, and therefore the X-gal cannot be cleaved into galactose. Okay, so therefore there will not be a blue precipitate. So there are 2 useful tools to characterize recombinant plasmids. The first one makes use of nucleic acids, and the second one makes use of proteins. Today, we're only going to talk about nucleic acid. Oh, there is a question, Azeri. No, there is no question. This is something from you. Okay. So, why do I go into all these things? Oh mon Dieu. Voilà. So, today we're going to talk about nucleic acids as tools to identify bacteria that are carrying recombinant plasmids. And on the 24th, we are going to see how proteins can be used to characterize recombinant plasmids. So, the first thing that you need to understand, first of all, and then remember, is the PCR amplification. PCR means polymerase chain reaction. It is a very, very powerful tool. And it is derived from the knowledge that we have gathered from the replication strategies. In the 80s, in the years 1970 or 80, researchers were learning how DNA replicated. And by and large, they developed a method whereby a double-stranded DNA could be very rapidly amplified, that is multiplied, so into two double-stranded DNA, and the two double-stranded DNA would give rise to 4, and the 4 would give rise to 8. So you can see that this is an exponential amplification. 1 will give rise to 2 in one cycle, and then 2 will give rise to 4. So therefore, from one double-stranded DNA, you can have massive numbers of the same DNA in 2 or 3 hours. So I have one question for you, dear students. Can you tell me, give me one example whereby the polymerase chain reaction has been used these last 3 years in human population? To detect something, so what was it?
The COVID-19.
Sorry?
The COVID-19.
Azeri, I did not hear.
The COVID-19, I guess.
The COVID, yes, absolutely, absolutely. To detect SARS-CoV virus 2, right? Sequences. So within 3 hours, so researchers knew the sequence of the SARS-CoV-2 virus, giving rise to the COVID disease, right? So they designed, you see those little red things? These are called primers, right? These primers will anneal, will link to extremities, and using a very special enzyme that we will see in the next slides, it will copy and make a complementary sequence of the blue DNA strand. And then there again, it will do the same job and it will amplify. So therefore, with only one DNA sequence, double-stranded, it only takes a very, very little amount. You can know in 2 or 3 hours whether you have a disease or not. This is widely used now to detect anything that has a nucleic acid sequence. For instance, the police uses this very, very often in all the cases where they have to identify somebody that has caused ill, that has killed somebody, or so therefore in our blood, in our fingerprints, in our hairs, we have DNA. So they come, they take a little bit of blood that has been left on the murder site, for instance. They go and analyze it, and they look through PCR and other genotyping methods. They try and identify the person that has this sequence. So how does this, so you need to remember this. It is very, very useful. It will carry you during all the years of your studies to the master, used every day in labs. So how does it work? So here you've got a double-stranded DNA, right? A blue and a red. So first, the PCR consists of a denaturing step at 95 degrees. So the denaturing step will destroy the bonding between the two strands, and these two strands will be separated. And then, because if we know the sequence, we will use primers. Sorry, this slide is in French. I could not find an easy one to show you. So it's a primer. It's a stretch, very small stretch of nucleotide sequence that comes here at the 3' end. And there is a very special enzyme called DNA polymerase, but this is stable at high temperatures, right? So first you have hybridization of the primer to the template. The template are the strands. That is the model on which has to be copied. So, and the primer, take it as an anchor. It comes and sticks here. The DNA polymerase is an enzyme that will elongate DNA sequences from the 5' end to the 3' end in a 5', 3' direction. You must have learned this during your first year. So you need to remember, okay? So you have two primers, and these will be used to extend using the thermostable DNA polymerase, right? And therefore, this is how you result with two double-stranded DNA. So that was the first cycle. And then the second cycle, it starts all over again. Each one of those double-stranded DNAs denatured, strand separation. The primer comes and fixes itself at each end, and it will give you the third cycles with 4 DNA, double-stranded DNA. Okay? So when you have these white colonies, it is very easy. If you take one of these colonies, or 10 of them, you make PCR on them, provided you have the primers that you are targeting. You know which primers have the corresponding gene you are looking for, you can make PCR, and within 3-4 hours, you can know whether these recombinant colonies are harboring your gene sequences. So this is a very powerful technique to screen bacterial colonies. So I have one question. Okay, just want to tell what we already know. Okay. Yes, thank you for asking this question. So the PCR steps, in order to get enough material, we need to make at least 30 cycles. So, and 30 to 40 cycles will take you about 3 hours. And these steps are very short. The denaturing step, 95 degrees, it can last for 30 seconds. This is a little bit longer, depending on the sequence of the primer and of the template. It will be for 1 hour, and this elongation step will be for, 1 minute, 1 minute, 1 minute. This can also be 1 minute to 2 minutes, depending on the sequence. So therefore, in 3 hours, you can get your result. Was this the sense of your question? Did I answer your question, Ario Bimo? Ario Bimo?
Of course, ma'am. Thank you for the answer.
Thank you. Okay. Thank you for asking. I hope that this will prompt other people to ask, other students. Okay. So this is one method, very powerful. There are many more methods, but I cannot go into all these details, right? So this is one thing that you need to remember. The PCR amplification method is a very powerful technique to identify very few DNA strand sequences that are present in a living organism. Right. So now we're going to go into something that is very useful because it is very difficult to see. Molecular biologists need to see things, right? Like we have seen color changes, right? Blue-white colonies, and we see colonies growing on antibiotic media, right? So this gives us some hints so that it can help us to eliminate those many colonies who are not harboring the gene. But now, let us go into a method that will allow us to even see much finer quantities. And this concerns there's another one question. No. I want to get rid of this. Voila. Okay. So this concerns the labeling. Labeling is when you will insert, while making a copy of a DNA or an RNA, a radioactive molecule. It can be a C, a T, a G, or an A. Normally, it's usually a C radioactive molecule. So you can label at the 5' end. You know that DNA, double-stranded DNA, are oriented 5' phosphate, 3' OH, hydroxyl, and here it's 5' hydroxyl, 3' hydroxyl, and 5' phosphate. So what do? We are going to remove this phosphate that is not labeled, and we are going to replace it by a phosphate group that is labeled radioactively. So therefore, this double-stranded molecule will become radioactive. So how do we do this? Of course, we have enzymes to do this. First enzyme is called a alkaline phosphatase. Alkaline phosphatase will remove the 5' phosphate and will remove the phosphate moiety group. And therefore, you will be left here with a 5' hydroxyl group at each end. Now that it has the hydroxyl group, you will replace this with a radioactive phosphate by using a kinase. A kinase is an enzyme that will add phosphate groups. Therefore, the 5' OH will become 5' radioactive phosphate. So this is extremely useful because these can be used as what we call probes. A probe is like a drone, right? That will go and look for its for the thing it's looking for. You know, drones go around and look for things, right? Drone may be looking at maybe a tree that is on fire, okay? And you in your forest, you cannot see it, but the drone will. So this probe is like a drone. It will go and look for the complementary sequence. And because you have labeled the extremities with a phosphate group, you will be easily able to see it because this radioactive element will print an autoradiography film. It's like when you make an X-ray, right? The doctors use films, right? And in the same way, each time you have a radioactive molecule, it will print on a radioactive on a photographic film. Next time I will show you a radiographic film so that you can understand what it is. Okay, so these are used as molecular probes and they are labeled, and because of this phosphate labeling, you will be able to see it. So this is the 5' labeling. The second method of labeling, which is much more used, is called random labeling. The random prime labeling is a method whereby you will incorporate, instead of labeling only the 5' extremities, you will label inside the DNA, every stretch of DNA all along the length of the fragment. So as usual, we start from the double-stranded DNA. We separate by heating. This means you denature the DNA, right? And then you include random priming. This means small pieces of nucleic acids, right, that are random. They don't have to be exactly, you don't have to know the sequence of your template. You just devise 8 to 10 nucleotide fragments, right? They are very short fragments, and these fragments will look for complementary sequences, and they will fix everywhere where they have complementary sequences. So this is the art of fixing primers to templates. So once these are fixed, we speak about annealing, then we get a DNA polymerase to work, right? And this DNA polymerase, in the presence of these 4 deoxynucleotides, of which one will be labeled, the DCTP. You can see it has a red thing here. This means the phosphate is labeled radioactively. So each time there will be a G, the DCTP will be incorporated. Therefore, you can see that this DNA will be elongated this way, right? And every time there is a G, the DCTP radioactively labeled will be incorporated. And on the second base, on the second strand, okay, you will extend the primers using DNA polymerase so that you will have lots and lots and lots of phosphate labeled nucleotides. And this method is, of course, more powerful than the 5' labeling because you have more specific activity, because you have more radio labeled phosphate groups within your DNA molecules. So what do we do with this? We will speak of it a little bit later. We will use these as probes, sort of drones, to look for complementary sequences. Okay? Now, we can mix this with PCR, and we will label the DNA in a much more rapid way, much more efficient, with very high specific activity by using PCR. So we will do the PCR exactly in the same way I explained to you in the first instance. But there again, we will include a deoxynucleotide that is radioactively labeled. So the steps are exactly the same. You've got a double-stranded DNA. You denature, right? And here are the 4 deoxynucleotides, one of which is labeled with P32, radioactive label. So therefore, when PCR starts, each time the enzyme will encounter a G on the template, it will integrate a DCTP. So you can see that this one will be highly labeled very, very rapidly, and you can use it for detection of very minute quantities. You see, everywhere will be labeled. So the thing is exactly the same as the first slide, except that it uses PCR. Therefore, the method is more rapid, and it gives rise to labeled molecules with higher specific activity. Okay? So every time you have those red rectangles, this means you have a DCTP that has been integrated, right? And therefore, this will be a highly radioactive molecule that will be used to probe your sequences. So we saw radioactive labeling. So you understand that working with radioactive elements is not very secure, right? We don't like working with radioactive substances in the lab. And to this end, in the late 80s, there has been something that has been developed. We can make exactly the same thing, right? Except that we will use a non-radioactive labeling. And this is called digoxygenin. So digoxygenin can bind to a deoxynucleotide sequence, right? And it will be exactly the same thing as what we saw in the preceding slide.
I'm sorry,
Using-
Prof. Jean.
Yes.
Before you continue the lecture, we have 3 questions in the chat box about the PCR. I'm sorry.
Okay, in the annealing process, okay.
Yes, the question is from Kiara Ali. She asked if one of the steps there is a temperature mismatch, can the process be repaired in the middle, or do we have to start over from the beginning?
Okay, first AR is responded. So, Kiara, I want to ask if at one of the steps there is a temperature mismatch. Temperature mismatch.
I think it means that the temperature is not correct.
Is not
Yes.
correct. Oh, this is a very long process to optimize the temperature. We work on when we optimize PCR, we will try different temperatures, right? Starting in order to optimize the annealing between the primer and the template, right? So if there is a mismatch, there should not be any mismatch. If there are mismatches, this means that the choice of the temperature has not been chosen correctly. Okay, So you will get very faint signals if your temperature, your annealing temperature is not good, or if the elongation temperature is not good. And usually this is optimized because you know the sequence, approximately you know the sequence of your templates, and you know the sequence of your primers. So for good matches, right, there needs to be a high percentage of GC sequences. The more you have GC, the more stable it will be. So there will be no temperature mismatch. The mismatch is between the nucleotides, right? Did I answer your question? Did you understand the answer, Chiara? Chiara. Ah, Chiara est partie. Azeri, is Chiara here or not?
Yes, she said yes, ma'am.
Okay.
But I think her question is, should we start over again from the beginning, or
Of course,
just
yes. Optimizing PCR is not an easy step. It takes about 1 or 2 weeks until you have a good temperature for annealing and for denaturation and for elongation. Okay?
Thank you.
So you're welcome. Tajuddin, I want to ask about the 3 main steps in the PCR, okay? What happens if we repeated the steps for over more than it should be? Then it will, you will lose time. You will increase your stock. But if 3 hours is essential, why do you want to wait for 6 hours? It's a waste of energy, okay? But nothing will happen. What is the function of DNTP during the annealing process? No, the annealing process is just the joining of a primer to a template. The DNTP, the deoxynucleotides, right, are used during the elongation steps when the polymerase adds the deoxynucleotide sequences. Yeah. You see, each time here during the denaturation, you don't need to have the DNTPs. Here you don't need to have the DNTPs during the annealing step. You only add DNTPs when you have to elongate using DNA polymerase. If the DNA polymerase does not have those deoxynucleotides, it will not be able to add the radioactive deoxynucleotides. Okay? Oh la la. What are all these questions? Has there help?
Yes, I think that's all.
Okay.
The next chat is about the attendance form.
Okay. I always get okay, good. So PCR labeling, is it okay to everybody? It's the same thing as PCR, except that the DNA polymerase will incorporate a radioactive nucleotide, deoxynucleotide sequence. Okay? the non-radioactive labeling is instead of the radioactive deoxycytosine, deoxy-CTP, you will add a dig molecule, right? That will be less harmful than the radioactive compound, but the principle is always the same. You have this dig is the label thing. It will allow to see you. Okay, right. Okay, we still have 10 minutes, so I can finish this lecture. So now we will see how we screen colonies using DNA hybridization, and this is where we will need the labeled probes. So here you have, after bacterial transformation, you have colonies growing on ampicillin on a medium containing the X-gal. Some colonies will be white and others will be blue. So you know that the blue ones are those that are of no interest, but you want to see if the white ones are all interesting to you, if they carry the sequence that you are looking for. So on your Petri dish, it is a very easy technique. You will put a nylon membrane on your Petri dish for overnight. And after one night, all these colonies will have been transferred to the nylon membrane. So the nylon membrane that was white or transparent will have exactly the same print as the original colony. You know that the blue ones are of no interest to you, but you need to know which ones of the white one will be of interest. So what do? We take the membrane, right? We will destroy the cell to denature the DNA of all the colonies. Okay, so therefore the DNA will be separated, the strands will be separated, right? And then we will add a probe. A probe is this, what I call the drone. It is like something that will look for a sequence to which it is complementary. So we add the probe on this membrane, we incubate it for the night, right? So if this is the probe, it will hybridize specifically to the colonies that are containing the right gene. So afterwards, you will wash the excess probe. You will place it on an X-ray film, right? And this will be the signal that you will see. It will be a radioactive spot. That is a black spot on your X-ray sheet. So this is a very, very sensitive way for you to detect the colonies that contain the strand, the gene you are looking for. So, if this is your colony, what do you do with it? You can't do anything with it. The only thing you can do is to get back to your original Petri dish and see where this spot is located, right? And if this is the corresponding bacterial colony, you will take it, you will treasure it, and you will cultivate it overnight. And now that you have your culture, you can work with it for 1, 2, 3, 4 years, or for how many years you want. Okay? So you know that within this bacterial colony, you have a plasmid that contains your sequence that you have inserted and that you are looking for. Okay? So I can understand that this is a difficult lecture, and therefore I will stop here and wait for your questions. Okay. Discussion. Right. Okay. So slide 2 reminds you that what you did in lecture 2. And this, today, we learned how to insert the sequences that we have been cloning last time, how to insert it into a plasmid and transform it into a bacteria. But of course, once you have all those thousands colonies of bacteria, you need to know what to do with them. You have to identify them. And therefore, you need to have tools to identify this. And to this end, there are 2 tools, color and antibiotic selection, right? So that's one thing. And a very sensitive thing is to label your DNA, make a probe, and screen your colonies by hybridization. That's all there is to the course. But unfortunately, because you are very young, you need to go back step by step. So now we have 7 minutes more, and I will answer your questions.
Okay.
I cannot believe it that you have understood everything.
Okay, if there is any question, please raise your hand or write your questions in the chat box. Jika ada yang ingin ditanyakan, silakan raise hand atau ditulis pertanyaannya di chat box. Terima kasih. Prof. Jin, before we continue to the questions from the student, I have a questions. Is it okay?
Yes, of course.
So in the last slide, you mentioned about the identifications of the DNA using X-ray by moving it to the nylon membranes. And then if we find out the results from the X-ray, we go back to the Petri dish. But my question is, if we take a long time to analyze under the X-ray, the bacteria in the Petri dish will grow, right? So if they're growing, it means they will change.
No. It does not take a very long time. They will not change because when you do this, you make prints. You make you will here. So here, when you have one Petri dish, in fact, you will have two. Okay, you make we have grids, and when we have this on the Petri dish, we will put exactly the same colony on a second Petri dish so that we can save it in the fridge. And therefore, when you have this radioactive signal, you can go back to your colony, because this one will not be good anymore because all the colonies will have been transferred to the nylon membrane. Do you understand?
Okay, I understand. Thank you.
We have to make two Petri dishes. Okay, when we have this, we take this one, we put on another Petri dish. We take this one on another This means we order the colonies. Okay, and the blue ones are not interesting, but it does not take very long. So they will not mutate because they are on antibiotic containing medium. You see? So there is a strong selection procedure. So these experiments last 3 days, not more. And when they are in the fridge, they will not mutate. If you put them in liquid culture, then they may mutate. That's a very nice question, Azeri. Thank you.
Thank you. Prof. Jin, there is a question in the chat box from Angkada Bima.
If the probe can identify YFG, like your last experiment, why we need to insert yes, good question. The probe needs to be labeled radioactively or non-radioactively so that it can easily identify the gene you are looking for. If it is not labeled, it will not be very easy for you to identify it because you will not see it. Angada?
Yes, ma'am.
Hello?
Could I ask to you to repeat it again with your answer?
Yes. So the probe will identify the sequence provided it has the complementary sequence. Okay, then it will match because it is a hybridization process. You know that double-stranded DNA, they are denatured, their strands separate, and then they will look for the complementary sequences. So why do we need to insert radioactive? If they are not radioactive, we will not be able to visualize them on a radio on an autoradiographic plate. You see, when you go to the doctor for a scan or for an X-ray, they use these special films, right? So these films are exactly the same films that we use, right? And radioactive elements or non-radioactive elements, they impact the film, and then it's like at the Photoshop, at the photographer. We reveal it, we neutralize it, and then we clarify it, and we can see where there has been the impact. Are you still confused?
No, ma'am, thanks.
If you
Thanks
don't,
for your answer.
okay,
What?
if you don't label either radioactively or non-radioactively, you will not be able to see it. For the next lecture, I will get you an autoradiographic plate. Sorry, I missed that. Thank you for asking, Anga.
Yes, ma'am. Thanks for the answer.
Very nice question. Any more questions, ladies and gentlemen? I understand that this could have been a difficult lecture. Yes or no?
Of course, ma'am.
But next time, it should be more simple, providing you work on the first 3 lectures, because you will need all the 3 of them, especially this one. Okay?
I got it, ma'am.
Okay, ma'am.
Ario Bimo, you have a question? No.
Maybe I have one questions for the last questions, I guess.
Yes.
Okay. In your opinion, it will be better if used the radioactive labels or the non-radioactive labels, in your opinion?
Yes. They are both very sensitive, right? You need to optimize it. I am a very old researcher, so in my time, there was no non-radioactive labeling. So I had to work with radioactive elements, right? With P32 and hydrogen and carbon-14 and etc. But of course, this was very dangerous, so we had to take lots of precautions. But now, for security reasons, we have the tendency of not using radioactive labels, because we don't want students to work with radioactive elements, substances, okay? And then for the environment, we need to get rid of them. It's too complex. So we work with non-radioactive labels, but both are as sensitive. It just depends on the optimization procedure. For the moment, I believe that you, when you will be going to work in your lab, you will use DIG labeling, non-radioactive labeling. It is very, very sensitive.
All right, ma'am. Thank you for
Can
that.
one of the professors, Arifin, what do you use at the UB in labs?
Oh, I use the chemical illuminescence one.
Of course,
I
yes.
didn't use
So,
the radioactive ones. So
yes, so yes.
I
you
use
would-
chemical illuminescence.
yes.
So is it easy? So and need not the radioactive installations.
Absolutely. So that's another probe, type of probe that we use. But I chose not to go into this because otherwise I will need more than 6 hours for the lecture. And your professors have given me 6 hours to do something extraordinary.
Yeah. Okay, but this is very great, I think. Yeah.
Yes, so if they don't understand, maybe they can get back to Afif or Arifin, Professor Afif or Professor Arifin. And if you need me, then you can send me a mail, Afif, if you need me.
Yeah.
But I think you
Yeah.
will be able. So I hope that this was clear. Afif,
Yeah.
je pense que c'était clair.
Is it clear? Pima?
Oui,
Pima.
le cours. Oui, oui, oui. Yes. Do you
It's very
think that
clear.
your students would have understood? Afif. Afif is gone.
Sorry, because
Yes, I think.
this time it looked like the call for praying.
For the prayers, yes. Okay.
So I think it's noisy enough.
Okay.
Do you hear? Do you hear?
Yes, I hear.
Yeah.
It's good.
Sorry.
It's good.
Yeah. So
It's
I mute.
it's
I
time
mute
for
my.
prayer. You need to pray because you have gone through this horrible disaster, right?
Yes, of
And-
course.
And so you are all in my prayers as well. Okay?
Thank you, Prof. Jin. I think Mr. Afif is not in the Zoom room anymore. I'm sorry. So,
it's
and the
okay.
time is already up. So
Absolutely.
thank you very much for
We are not
your.
going to do overtime.
Okay, thank you, Prof. Jin, Kallerhoff, and the
Thank you.
participants.
Thank you to you. Thank you.
Thank
you.
you very much.
Bye-bye. Bye-bye.
So
Thank you very much.
before leaving, Prof. Jin, I shall
Yes.
told you. I shall tell you that for the next meeting, maybe I can't join you because I have a trip to other. So I'm very sorry.
Okay.
The next,
But-
But
Yeah, if
Azeri
I
and Dr. Afif will join you.
Okay, good.
Yeah, and also Prof. Arifin, in
Yes.
the last meeting,
Yeah.
our head of department will give closing statement.
Okay,
Okay.
thank you, Afif. Yeah.
Mm.
Thank you very much.
Okay,
Yeah,
thank you,
so-
Jin.
Thank you very much,
Bye-bye.
Jin.
Bye-bye.
Bye-bye.
Bye.
Thank you. Bye-bye.
Makasih Pak Afif.
Ya, Pak.